Are there genetic tests for anhedonia in schizophrenia? If you have ever wondered about the connection between genetics and anhedonia in schizophrenia, you are not alone. Anhedonia is a key symptom of schizophrenia, marked by a reduced ability to feel pleasure. While it is a significant aspect of the disorder, the question arises, can genetics help us understand it better? Currently, there are no specific genetic tests available to diagnose anhedonia in individuals with schizophrenia. However, research indicates that anhedonia has a genetic basis. Studies have shown that many genes contribute to the risk of developing anhedonia, which is also linked to schizophrenia and other mental health conditions. Genome-wide association studies have identified various genetic locations associated with anhedonia. This means that multiple genes, each having a small effect, play a role in its presence. These genetic factors overlap with those involved in schizophrenia, major depressive disorder, and bipolar disorder. Researchers often use polygenic risk scores to measure the genetic risk for anhedonia. These scores combine the effects of numerous genetic variants related to the condition. Interestingly, higher polygenic risk scores for anhedonia have been associated with changes in brain structures that process reward and pleasure. Areas like the orbitofrontal cortex show alterations linked to anhedonia symptoms. Despite these findings, the genetic landscape of anhedonia remains complex. Most current studies assess anhedonia at a single point in time, which may not capture the full picture of genetic risk throughout a person's life. While polygenic risk scores can indicate a higher vulnerability, they are not diagnostic tools. They cannot predict whether someone will experience anhedonia or schizophrenia with certainty. In the context of schizophrenia, anhedonia is viewed as a negative symptom, reflecting underlying genetic and neurobiological vulnerabilities. Family studies reveal that relatives of individuals with schizophrenia often show higher levels of trait anhedonia. This supports the idea that anhedonia can serve as a marker for genetic risk. However, no clinical genetic test exists specifically for identifying anhedonia in schizophrenia patients or those at risk. Instead, healthcare professionals assess anhedonia through clinical interviews and standardized rating scales. As research continues, the understanding of anhedonia's biological basis and its relationship with schizophrenia may lead to more targeted interventions in the future. For now, genetic testing for anhedonia remains a tool for research rather than a method used in clinical practice.